Good afternoon, good evening. I think this is a great venue, and I want to first and foremost thank Nana, Rory, Beth, the whole TSRC staff. I think we should really give it up for them. So we are going to talk about advancing science through diversity. I initially wanted to title this, How Xenophobia Weighs You Down. Xenophobia is the fear of the other, the fear of things you don't know. And diversity is about accepting the things that you don't know and thinking about how that actually can make you and others better. That's exactly what I've been doing in Telluride. Anyone recognize this little ledge? Anyone? Does it look familiar? So where is it? Where? Palmyra? Sort of overlooking Lizard Head, right? Is that Palmyra? No, so this is on the Lizard Head hike. And we did that in 2003. Blackface, exactly. And the reason we enjoy coming to Telluride is because when we start walking through your mountains, when we start walking through these landscapes, it gives us the opportunity to talk to each other in a way which is different than the way that we normally engage. And that allows us to rethink our problems. Sometimes even the landscapes of the surfaces remind us of the energy landscapes of the problems that we're thinking about. So that even inspires us to talk about things like, how do you go from a reactant to a product through the saddle point of the mountain, right? You don't want to walk any extra. And that makes it very easy to engage students and each other in those types of metaphors and translate it into different types of science that we engage with others. We may have seen the fact that we had some pictures earlier about really strange particles, molecules. So we've been organizing a workshop since 2001, every 2 years, on complex environments. And so I'd love to talk with you about that some other time, but today we're going to talk about diversity equity. Okay, so diversity equity. What do I mean by diversity equity? Diversity, first I have to define, and so I'm going to define that in a second. Well, how about right now? Diversity is basically the inclusion of the other. Okay? And that can mean just accepting. I know it's crazy. Most of you, this is Telluride, so you're probably what? Democrats? A little on the liberal side? And I know how hard it is to accept someone who's a Republican, but they're people too. And so we want to be able to accept each other, regardless of our beliefs, regardless of where we come from. Now, some things maybe where the person was born. That adds color. I love going to Siam and having some Thai food, right? And it adds to the breadth of the kind of food that I eat. And they didn't pay me to say that. I don't know if I'm allowed to mention that. And the idea is that somehow we are strengthened by including others. And in particular, I want to point out that we're strengthened by including gender diversity, race, ethnicity, disabilities, and lesbian, gay, bisexual, transgender, queer, intersex, and questioning. Plus any other type of gender identity you wish to have. Because if we don't accept each other, then we are restricting the kinds of people we get ideas from. And that's exactly the problem we have in science. If we only include one set of people thinking about very difficult questions, we're not going to solve the problems that are facing us in energy, in water, in health, and other things, which are often the types of talks that you hear about in Telluride. So I want to talk about diversity equity, which means we want to make it fair, equitable for everybody, regardless of their background, to have access to being scientists and therefore contributing to society. You might wonder, why are you talking about that in science? Well, it's actually timely because we're talking about this in the national conversation. Even with respect to women, gender, as much as we've done in terms of the inroads, getting women to be equitably represented at various levels, still there are issues like whether you belong or whether you should lean in or lean out, and does it depend on your gender, etc. That's still not well resolved. Or you can ask about Alan Turing, who essentially helped England win the war and yet died because of his sexual orientation, and which he could not control. There are countless individuals who can contribute, and yet just announcing that you're gay makes frontline news, even if you're like a nobody soccer player. I mean, so clearly our society hasn't come to grips with that. Or you look around the table of the people who are underrepresented minorities, and you find them at the top of their profession, and yet they're not equitably represented throughout. Or you talk about disabilities, individuals that can contribute to society in ways that no one can, and yet if we ostracized them and kept them away, science and society would not be advanced. So diversity, equity in science is important because we need to have that thought process, that thought power in order to advance science. And so you might say, well, it should just be social justice. But here's the bad news. As much as I believe in social justice, anybody here believe in social justice? Social justice was not going to get me the funds to advance science, diversity, equity in science. Because this is not about social justice. This is about competitiveness and excellence. That is what drives science. And the truth is, we are more competitive, we're more excellent, or we can achieve excellence by being diverse. So we're not talking about any kind of affirmative action. We're just saying, if you make the playing field level, the best will rise. And the problem right now is that the playing field is not level. So first thing I want to do is convince you about the fact that the playing field is not, in fact, level. If you look at and I'm a chemist, so I focus on chemistry, okay? And if I look at the numbers in chemistry, in the top 10 chemistry departments, according to research expenditures, we can quibble about how we rank them, and let me tell you, we do a lot. But in the top 10 or top 25 or the top 50, the top 75, women are about 17%. I think that's a little different than the demographics of our world, of our country, right? If you look at the male to female ratios across ranks, assistant, associate, and full, it's actually better among the assistant ranks, which might make you think, oh, life is great. You just wait. Assistant professor's lifetime is about 6 or 7 years. Associate professor's lifetime to full, that should be about 10 to 14 years. We'll start getting closer. But we've been about that percentage for the last 20 years. We've gone from about 11% to 16% over 20 years. There's a significant attrition, comparatively significant attrition of women in the sciences as assistant professors in comparison to the men. Okay, so that's why it hasn't rolled over equitably. All right, so that's one example. We can look at the demographics of the students. So if we look at 2010, of the 2,300 PhDs in chemistry, 37% were female. Wait a second, it was about 16% of the faculty? That looks like there's a drop from PhDs to faculty, even for the assistant professor level. Among the U.S. citizen or Puerto Rican PhDs, that's about 57%. 0.4% are Native American. That compares to anywhere from about around 1%, because these are sometimes lumped together in that. 10% Asian, African American 4%, not 12.6% in the national demographics. Hispanic 4.8%, not 16.4%. There's a significant difference in those percentages from general demographics to PhDs, and the drop-offs go even further when you go to faculty. Meanwhile, let's think about some of these numbers, okay? We've got about 6,000 bachelors in chemistry. That translates to about 1,000 PhDs. So 1 PhD for every 6 bachelors. Then you take the PhDs to the faculty members, and I'm going to give a faculty member about a 25-year lifetime. She might have 25 years because, well, she didn't get tenure. She died. But maybe she lived for 50 years, or he lived for 50 years, right? It's a he or she. Let's go back. And so about 25 years is their average lifetime, so about one professor for every 17 PhDs among the faculty in the top 50 chemistry departments. And so your odds, if you're a bachelor's level chemist, of being a faculty member, if that's your goal, in the top 50 chemistry department is 1 in 100. 1 in 100. That's the intellectual 1%. All right? And let me ask, so if you're in that, trying to be in that intellectual 1%, how do you know you're in that intellectual 1% graduating from college? So I like to say that I was extremely foolish to think that I could beat those odds leaving my bachelor's degree. But that was exactly what I did. And we're expecting that enough individuals who have, in some cases, very competitive alternative jobs to take that leap of faith. So any difference, any inequity in going from a bachelor student to a faculty member that will preferentially make it harder for you to be a faculty member than someone else, you're going to be afraid of. And I'm going to now try to convince you that there are indeed diversity inequities in this process, which make it, at least up to now, which make it so that underrepresented minorities, women, were making the choice not to take that jump. And so all we need to do is fix those inequities. That's all. Is fix those inequities, and then we'll be able to level the playing field, and then we'll get the numbers we want, equitably in comparison, without doing anything, sacrificing no competitiveness, no excellence, and in fact, gaining excellence, because now you have a more diverse and qualified set of faculty members. So it really is about excellence and the meritocracy. Now, faculty members live and die because we have a faculty that is the best, and we hire the best. And because we do it well, and we know what good is, we always hire the best. So let me see how well we are at seeing things when we're looking at them. So everyone here, you can see this red, the blue, the green curves, right? Everyone sees these three colors, blue, red, green? Everyone, right? Sure. There's the blue color and there's the green color. Wait, blue, green? Here's a line. Follow that line. Oh, he must be playing some games with us. Follow this line right here. It's green, blue, blue. Wait a second. The blue and the green line are exactly the same. Your eyes are deceiving you. But it was right in front of you, the same application. How did you not tell that they were the same? Let's look at another example. Now, I have to ask a question. Is anyone in this room a black belt in Taekwondo? All right, my son is. Anybody else a black belt in Taekwondo like me? And because if you're a black belt in Taekwondo, you're going to really know the answer. So I have to be very careful. Because, so if you're black belt and you're an expert, please do not say the answer. Here are two individuals, the one on the left and the one on the right, A versus B. And I want you to think about who is about to score the next point. Okay? Now, everyone agreed that it's not the same, that no one else is an expert besides my son, right? So my son can't say anything. When I say 3, I'm going to want all of you to say A or B. Does that make sense? I'm going to say 1, 2, 3, and you're going to say A or B all at the same time. Ready? Yes. Ready? Yes. Okay. 1, 2, 3. A. Wow! You guys are experts! Because indeed, the red one wins. Preferentially, whichever one of these two is wearing the red gi gets the point if you do a survey. And that's what they do. That's what Fred Mice does. It shows that if you show the same figures and you just change the color, whoever one was red, A gets the point. You are all perfectly good at liking red as being more aggressive and winning the point. So you would hire the person coming into your office wearing red if the job required a go-getter. And that was completely arbitrary. All right? These are the kinds of barriers that we have to achieving diversity equity. We are all biased. No crime in being biased. I'm biased. We're all biased. I said red too the first time I saw that. The key is knowing what your biases are, knowing how you are applying them in choosing the best, in choosing the people. And the problem that we have is that we have not been aware of the biases either in the making the decisions or in supporting the individuals once they're in the system. And there are a lot of barriers. And this is the problem, that in 20 minutes I had to tell you about everything. So I'm not going to tell you about all of these barriers. I'm just going to whet your appetite. Implicit or conscious bias is the one that I gave you a lot of detail. We all have implicit bias, and that implicit bias is a diversity inequity that we apply in making our choices. We do that in faculties. There's a number of things like schemas that you have a pre-existing place that if you see someone, you place them in that schema, and therefore you expect they're going to act in a certain way. A number of things. Lack of universal design. Has anyone here ever used an assistive device? Have you ever woken up with an alarm clock? Have you ever used a shower? You're all using assistive devices. They're tuned to your particular needs. If we have the appropriate assistive devices, we're on the same level playing field. I couldn't wake up with that assistive device, the alarm clock. Therefore, I'm able to make my meetings on time. There are a number of others, things like solo status. There is plenty of evidence that shows that individuals act differently when they're in a room of people that are all different than them because they're pressured, except if they're a white male. So if the white male's in the minority, he still acts the same way. But I don't know why. But anybody else that's in the minority in a room, they act differently and they tend to underperform. If I tell you before you are going to do an exam that your exam, if I prime you and tell you that your exam is somehow contingent on representing your race or your identity, if you are Asian, you tend to overperform. If you're Hispanic, underrepresented minority, or female, you underperform. If I've primed you. And I can prime you just by you coming in, and this would be stereotype threat, having made sure that the women's bathroom is on the 4th floor, the men's bathroom is on the 1st floor, and you're all here on the 1st floor. Just that stereotype threat primes you in knowing that you as a woman are second class. Yeah. All right. So there's a number of things, if I had more time, we could go through and look at just the large number of different inequities that exist in the system, despite all of your best intentions. I'll mention one more. I don't worry about the color of someone or whatever. No matter what they are like, I know I will always act the same. That's the color blindness policy, right? Except it completely fails. Colorblind policies, supposedly colorblind policies, tend to be more color conscious and result in more diversity inequities than not having it at all, which is counterproductive. But again, recent data done in the last couple of years, I'm just reporting the facts by social scientists, have given us that idea. So what did we do? We have the idea, you build it and they will come. So reading between the lines, diversity is a planned event. If you want a diverse faculty, then you have to plan for it. How do you plan for it? By ensuring that there are no inequities in the process of bringing these individuals. And so I have a team, the Open Chemistry Collaborative in Diversity Equity, and we've been funded since 2012, although we started earlier than that, to work with the chemistry departments around the country to look at their policies and procedures so as to help achieve the demographics of the nation. So to help them find the best minds. So we're on the same page. We want the best departments. We don't have the best departments right now because they're not diverse. Very simple. And so what do? We collaborate with social scientists. We think about diversity of all types because it's not just one type of diversity that's an issue, it's all types. And so we were the first group doing this that included sexual orientation, LGBTQIQ, because we recognized even just years ago, it's been very timely now, that was also lagging behind. We disseminate, we host national meetings. The department heads of these universities, like from Berkeley, Chicago, Northwestern, Georgia Tech, San Diego, they come to this meeting and we talk to them about what the policies and procedures are, what their effects are, and how they can change it. And we continue that conversation between these biennial, that means every 2-year meetings. And in addition, someone told me we're a watchdog. We are measuring the demographics of the departments and we're publicizing them. So if your demographics aren't good, your department doesn't look good. These are the members of my advisory board. I'm proud to know that say that several of them are social scientists. We include the same type of diversity spread that we're aiming for in our board. If we even have a white male. What can you do? Diversity is a planned event. You can think about diversity. You can think and learn about the kinds of biases that cause you to make biased decisions, and then think about what works for you to make unbiased decisions. How do you self-correct for your biases? By the way, sometimes biases are really good. Any scientist in the room will tell you that their instincts for the best science is absolutely correct. The only problem is that once you ask any pair of them about their biases for the best science, somehow there will be differences. And therein lies the problem. You have to recognize that your biases have helped you, but that you need to be cognizant that you have to be unbiased to help everybody else. And with that was advancing science through diversity.
Thank you very much. I'm really glad to be here. This is my first time in Telluride. And as you can see from the slide, this is a slide of Notre Dame. The terrain is a lot different where I come from. And so it's been a significant, we're also at sea level, so it's been a significant adjustment here at Telluride. But I think I'm over the worst of it. So I'm delighted to be here. It's a beautiful, beautiful place. And the science already has been really great. So I'm looking forward to the rest of the conference and talking with you as well after the talk. So what I want to talk to you today about is bacteria, and specifically the relationship between humans, the evolving relationship between humans and bacteria. So, and the bottom line for bacteria, that this is my first, if you don't remember anything else, remember this slide for the talk, is that bacteria are small and simple, but they're also quite diverse. That sort of echoes some of the themes from Rigoberto's talk. And they also, because they can reproduce very quickly, that means also they can adapt to new environments very quickly. And something that's been going on now for about 70 years since the development of antibiotics is we've been waging a war against bacteria. And what I'm hoping to convince you of today is it's a war that we cannot win, and that we really need to adopt a very fundamentally different strategy for interacting with bacteria in order to peacefully coexist with them. So that's really the bottom line here, is we need basically to achieve détente rather than continuing to fight this war. Okay, so bacteria are all around us, and I think there's been a lot of publicity recently about bacteria in relationship to humans, and that's what I'm going to go over here. And I realize that some of this is probably background for many of you here, but I want to make sure we're all on the same page about where this starts. So bacteria are all around us and in very large numbers. So if you can think about your average citizen, is actually basically a walking bacterial transport system. And this is because humans consist of about 10 times more bacterial cells than human cells. And this surprised a lot of people when it first came out, especially because we look like humans, we don't look like bacteria. But this is because bacteria are an individual bacterial cell is much smaller than a human cell. And so as a result, if you kind of scrape together all those bacteria into a pile, it would be about the size of a half-gallon milk jug. Okay, still quite considerable, considering we don't really see them on a daily basis. Okay, so for example, there's about 100,000 bacteria in a square centimeter of human skin. So I'm going to warn you, this might creep you out a little bit, and that's kind of part of the point, is to creep you out a little bit. My husband gets really, really creeped out about this, so he always feels the need to wash his hands after I talk about this. Okay, and the mother load of bacteria are in our gut. So this is what's called our gut microbiota, or gut bacteria. So our gut bacteria are numerous and also very diverse. And they vary a lot from individual to individual. And there's actually an enormous program going on right now that's funded by NIH to correlate an individual's, their genetic makeup, so your human genes, with what sort of bacteria colonize their gut, their skin, various other locations on the body, including belly button, which I thought was a very interesting sampling site. Okay, and one of the very first findings that's coming out of this focus on the gut bacteria is that our bacteria in our gut contribute essential features to our metabolism. So what's been shown recently is that if we make radical alterations to a human's gut bacteria, that we can have, in sometimes cases, severe effects on their ability to digest food, and that can lead to things like obesity and also digestive diseases such as Crohn's disease and inflammatory bowel disease. Okay, and this is, I think, just the tip of the iceberg here. The others with a question mark after it is because this is really the Wild West, right? We learned Telluride here, which is also the Wild West. This area of research is really the Wild West. It's so recently that we've appreciated what a significant component bacterial metabolism is to our human metabolism that I think we still have much more to learn than we actually know yet at this point. Okay, and obviously bacteria are not just attached to us, they're also all over the place. So your average shovelful of soil includes billions of bacteria. And some of that's also coming from us. So about 60% of the dry weight of our feces comes from that gut microbia. And I think we should all just pause and rejoice here in my decision to not infect you with, or not subject you to my cartoon PowerPoint rendition of feces. Okay, I restrict myself. Okay, so the fact that we have this gut microbia that's clearly contributing very strong positive influences to our metabolism, and yet we all get kind of creeped out at the idea of all this bacteria on our skin and in the soil and everything we touch, I think reflects this duality we have right now between bacteria, sort of the good versus the bad, the good, the bad, and the ugly, because some of them are quite ugly as well, as we'll see in a moment. And I like to illustrate that by this slide here. So this is the good side of bacteria. So we have a long-standing positive relationship with bacteria in the form of yogurt. So humans have been culturing yogurt for about 4,000 years, right? And everybody, I mean, you should all love yogurt. It's wonderful stuff. And talk about positively contributing to your gut microbial balance. There's lots of good things from yogurt that have been shown, again, in controlled clinical trials to positively contribute to good bacterial health in your gut. And conversely, there's antibacterial soap, which, as Beth alluded to, is something that's been it's a very interesting contrast here, right? Antibacterial soap, the idea here is any bacteria that's on our skin we want to kill, right? Every time we wash our hands, we want to kill bacteria. We don't want to have any nastiness under our fingernails and so forth. And so this duality, I think, is really created this situation right now where we need to find out a way to kill the bad bacteria, but really nurture and foster the good bacteria, both in things like foods and also in our gut. Okay, so I wanted to talk a little bit about why bacteria are always going to win. So basically why this idea of a war on bacteria is doomed, and why we can apply all the soap in the world and we're never going to win, okay? And have all the antibiotics we want of the current kind that we use, and we're never going to win. So basically this is related to exponential growth. So bacteria grow exponentially just like everything, so they're able to double, and the point is that they, as opposed to us, can double incredibly quickly. So for a human, it takes 9 months to produce a baby, and we get one, or maybe two or more, right? But usually just one. Okay, and for a bacteria can produce one offspring every 20 minutes, and as soon as one is born and becomes 20 minutes old, it can produce a daughter, right? So it's both able to divide very quickly, and also it matures very quickly as well. And so I tried the other night before I came here to say, okay, well, what about if we had 9 months of bacteria doubling, how many offspring would we get? And the numbers are just staggering. It's one of those ones that the exponent is so large, it's meaningless. So I thought instead I'd approach it from a different perspective and say, okay, the Earth's population, human population, is about 7 billion people. So how long would it take bacteria, a single bacteria, to produce 7 billion progeny? And so that's very simple to do, right? So you can do all the doublings here, and here you get to even more than 8.5 billion people, and that took 33 doublings, okay? So if we can get to more than 7 billion progeny, 33 doublings, if we double every 20 minutes, that's going to take 11 hours, right? So that's really staggering. I think even for somebody like me who thinks about that a lot, this is already just a staggeringly fast ability to produce offspring. Now, this is obviously, this idea of a 20-minute doubling time, that's under sort of ideal growth conditions. So abundant food, no predators, no challenges to the environment like extreme temperatures or weird chemicals and things like that. And that's obviously not always going to be the case. But you can imagine, if you've got 8.5 billion offspring, you can sacrifice a few to a poor environment. And also, every time you replicate, you introduce the chance of having mutations, some of which, again, are going to produce offspring that aren't going to make it. But every so often, you'll get one that'll do better in whatever challenge you get. And that's where this idea of resistance comes in, that every time you double, every time a cell replicates itself, you introduce a chance for mutations. And it's just pure plain statistics. When you have this many chances to get better, you're going to get better. Okay? And that's why we're never going to win. Bacteria are always going to be able to outmaneuver us just from the numbers alone. Okay. Okay, so they've got a short gestation time and early maturity. And so again, getting back to this idea about what bacteria need to do, not in ideal growth conditions, but as pathogens. Let's start thinking a little bit about pathogens. Again, so this is the idea that we have lots of bacteria, and we don't really mind so much. Maybe we can get comfortable with the idea that there's bacteria in the soil, and bacteria that are helping us in our guts, and even on our skin. But what about these pathogens? What about the bacteria that can actually make us sick? So what are they facing, and what maybe of those things that they're facing can we exploit to try to make our bodies less hospitable environments for them to take up residence? So the bottom line for any pathogen, a bacterial pathogen, is it's got to invade our body. It's got to basically set up housekeeping. So it's got to find some sort of ecological niche. And that's true whether it's a human pathogen or whether it lives in the soil or something like that. It's also got to evade the immune system or whatever kind of drugs we might throw at it. So it's got to be able to hide from the immune system and neutralize any sort of antibiotics. And it's also got to be able to live and reproduce. So it's got the end goal for bacteria is to reproduce, right? So all they live for is making another copy of themselves, and to do that they need to recruit nutrients. Okay, so let's talk a little bit about how these play out in bacteria. Okay, so first of all, actually some general features about bacteria and how we combat them right now. So when we talk about drugs and antibiotics specifically, antibiotics are designed to exploit something that bacteria need to do that hopefully human cells don't need to do. And therefore, the drug that we design is going to target that thing that bacterial cells do and human cells don't, and therefore it will be toxic to the bacteria but will not be toxic to humans, so we'll have minimal or hopefully no side effects. And so this is a generic sort of picture of a bacterial cell. You can't tell from this picture whether this is a pathogenic bacteria or not, but a couple things you can see that are different about this cell, or this type of cell, versus a human cell, are these guys circled in purple here. So bacterium have this structure called the flagellum. Many of them do, not all of them, have this long tail-like structure that they swish back and forth like a fish tail to enable them to propel forward and move. They also have these shorter appendages called pili, and humans, we have something that's also called the pili, but it's not at all the same. And they also have this interesting cell wall structure, which is basically this rigid husk around the bacterial membrane that enables them to withstand osmotic shock. Okay, so that if they lose that husk, or if it becomes destabilized or it gets a little hole in it, then the cell might rupture. Okay, so those are all things that are currently used by either our immune system or by current antibiotics to target. So the most famous of all antibiotics are the penicillins, the beta-lactam antibiotics, and those target this cell wall biosynthetic process to try to reduce the bacterial's ability to produce a cell wall. And they're obviously very effective. So one of the things that I think is interesting about this picture is you can't tell here any distinction between a pathogenic bacteria versus a bacteria that lives in your gut. And so I wanted to also show you a bunch of pictures of bacteria as we start thinking about, again, what distinguishes a pathogenic bacteria versus a good bacteria, and how we might exploit those differences to try to design better antibiotics or better methods in general beyond even antibiotics to combat bacterial infections. Okay. So here's an electron micrograph of E. coli. This is a laboratory strain of E. coli, but honestly, you wouldn't be able to tell if it was a pathogenic strain either. They look basically the same. They look like little rod-shaped bacteria. So lots of us are familiar with E. coli from the news because of things like infected hamburger and spinach and so forth. So those are commonly contaminated by E. coli. Here's another human pathogen, H. pylori. This causes stomach ulcers and stomach cancer. So it takes up residence in the stomach. It's got special proteins within it that can enable it to live in the very low pH acid environment of the stomach. You can see it's got very prominent flagellum. This is a non-pathogenic bacteria. This is an unclassified cyanobacteria. Cyanobacteria are really cool. So they are actually green. They possess chlorophyll, and they can do photosynthesis. And this is an enormously important class of bacteria that I bet if there's any picture I show up here of a bacteria that you've not heard about before, it's probably the cyanobacteria. So they're sort of the underappreciated bacteria in the bacterial world. So they constitute, so first of all, these bacteria, because they're doing photosynthesis, just like plants, they produce oxygen as a byproduct of photosynthesis. And so what this means, there's so many of them. Again, remember, bacteria, incredibly large numbers of them. They're doing about 20 to 30% of the world's photosynthesis and producing about 30% of the world's oxygen. Okay? All right. But they're not pathogens. These are not human pathogens. Here's another, I'm not going to try to pronounce this. This is a variety of bacteria that's actually not pathogenic to humans. It's pathogenic to other bacteria. So any other bacteria that it encounters, it will actually gather around and eat. Okay? Break down and eat. So this is a bacterial pathogen that is itself a bacteria as well. Yes. Okay, here's another spiral-shaped bacteria. So these guys are all kind of rod-shaped and similar in that shape. There's a spiral-shaped one. This is the class of bacteria that causes Lyme disease and Lyme disease. Okay, here's Campylobacter Jejuni. So this is the major cause of foodborne illness in the United States. So if you get food poisoning from eating in a restaurant that didn't prepare its food correctly, it's probably from this bacteria. And you can see it kind of like these two guys share that they have this sort of spiral-shaped structure. And there's others that are small circles. So this is a very famous bacteria, Staph aureus. So this is the primary cause of hospital-acquired infections. So there's major problems now with resistant strains of this bacteria that are basically resistant to all classes of antibiotics that we have. And here's a related circular bacteria, or related in the fact that it's also kind of spherical, but instead of forming these grape-like clusters, this forms long strings, kind of a beads on a string structure. This is Streptococcus, and this is the bacteria that causes strep throat. And the exact same bacteria is also the flesh-eating bacteria and causes scarlet fever as well. Okay, so wide variety of bacteria. But again, they all share some common features that we can try to exploit for creating novel strategies for combating bacterial virulence. I wanted to mention one last guy. This is another type of cyanobacteria, a specific one called Prochlorococcus. This is found in the open oceans. It's very temperature sensitive. So this bacteria here is doing 50% of the photosynthesis in the open ocean, so 50% of the oxygen production in the open ocean. It's very temperature sensitive. So this is the one reason why I worry about global warming. Okay? Because if the global oceans warm up by just a few degrees, something's going to happen to these guys. Maybe they'll adapt and change, or maybe something is going to take over in their place. I don't know. So just food for thought. Okay. So pathogens. So again, still from these pictures, we can't distinguish a virulent strain of bacteria from a non-pathogenic sort of laboratory strain of bacteria. And my own lab, like Beth mentioned, works on bacteria, and we work on laboratory strains of bacteria that are very benign. So they're sort of like these civilian models of these Humvees. So Humvees are made right in Mishawaka, right down the road from Notre Dame. So the civilian models here, you can see that they look kind of tough, but they're not really equipped for warfare. And in contrast, if we look at what pathogenic strains have, they're the same basic vehicle, right? So they still have 4 wheels, they still have seats, steering wheels, and all that sort of stuff, but they've clearly got a lot of extra stuff that enables them to be pathogenic. And this stuff is also itself very diverse. So there's like big guns here up on top, this guy, and so this might, in the bacterial world, might be equivalent to something like this type 3 secretion needle. So these type 3 secretion needles are found in a class of bacteria that actually have 2 membranes, so they're bounded by 2 membranes, and they construct this elaborate structure made up of all these different proteins, all these different colors and letters are different proteins, that spans both membranes of the bacteria and the space between the bacterial cell and a human cell, and creates this syringe or needle-like structure that can directly inject bacterial proteins into the human cell. Okay, and then can use that as a strategy to recruit nutrients back. Okay, there's also smaller guns. So this might be an example of, for example, the kind of proteins I work on, which are autotransporter proteins. So they're called autotransporters because they seem to have this unique ability to be able to flip themselves across the outer membrane to get out to this outer surface where their function is needed. And these are the most common type of bacterial virulence proteins in gram-negative, which is just a class of bacteria. So again, if we think about these 3 things that all pathogenic bacteria need to do, they need to invade, evade, and recruit nutrients. I wanted to use just a couple examples of autotransporters as examples of how to get by those strategies. Okay, so we need to find a home with sufficient nutrients, and we need to avoid antibiotics and other ways to kill. So if we think about finding a home with sufficient nutrients, so for example, a big problem with bacteria who infect humans is getting iron. So it turns out that humans have developed the ability, probably to control bacterial infections. We control iron very, very tightly. So iron is not floating around. Okay, so the way that we have used 3 different strategies to control iron. The first is that we hold on to iron with proteins, so things like hemoglobin that we use for transport of oxygen, and another protein called ferritin. So we hold the iron inside cells and we keep it attached to proteins. The other thing is that we live in an oxygenated environment and our blood is oxygenated, and that creates an oxidation state of iron that's actually pretty insoluble. Okay, so that's not very easy for bacteria to grab hold of. And also, circulating in the blood is another protein called transferrin, which has an enormously tight hold on any remaining iron. Okay, so this is a dissociation constant that means that basically you put 1 molecule of iron in a swimming pool and this protein is going to be able to find it and grab it. Okay, so when bacteria enter a mammal like a human, they sense this iron depletion. So they'll start losing iron from their essential proteins. And they'll start responding to that by upregulating the production of other proteins to try to get iron back. So, for example, they produce a class of small molecules called siderophores. These basically compete with transferrin. They have an even tighter binding affinity for iron, so they're trying to grab up any sort of free iron. And then there's other proteins that can strip iron from other proteins, like hemoglobin. So this is an autotransporter protein called hemoglobin protease. So its job in life is to go out into human cells, specifically red blood cells, chop up hemoglobin, grab the heme, and bring it back to the bacterial cell. Okay? All right, so that's one autotransporter protein, and what I'm going to show you just at the very end here is a little gallery of a couple others. So this is the same guy I just showed you, E. coli hemoglobin, that's involved in iron recruitment for pathogenic strains of E. coli. Laboratory strains of E. coli don't have this protein. They can't produce it. Here's another protein called Ig protease. So Ig stands for immunoglobulin. This is a component of your immune system. So the function of this protein is to go out and chop up antibodies so that your immune system can't detect the presence of the bacteria. Okay. Here's 3 other ones. These 2 my lab works on. This is a protein from Bordetella pertussis, which is the bacteria that causes whooping cough, primarily in small children. It's called protactin. It's involved in adherence, so it helps bacterial cells stick to the surface of human cells. That's all it does. Okay? But again, that fulfills one of these 2 roles here. Bacteria have to find a home, and obviously you have to be able to stick to your home. You have to be able to find sufficient nutrients, and you have to be able to avoid antibiotics and other killers like the immune system. This is a toxin. It pokes holes in mammalian cell membranes. And this is another, I'll skip this one since I'm running low on time. The one thing I did want to point out is that these proteins are already being used to combat bacterial virulence. So Protactin, the one from the bacteria that causes whooping cough, is already a member of the 5-component acellular whooping cough vaccine. So my son, for example, and any of you who have children who are younger than 20 have probably been injected with this protein. And the nice thing about it is it raises a very strong immune response, so that if then your child ever sees that Humvee studded with this protein out on the surface, it'll swoop in immediately and kill it before it can start to grow and propagate and produce all those billions of progeny cells. One of the reasons it's thought that these proteins become such great vaccine candidates is because they all seem to have this beta helix structure, this structure shown in blue, which doesn't seem to be really related to the function, which is really more the parts that are in red, and is also not used in human proteins. So again, those of you who know something about the immune system know that the immune system has a very sensitive response to things that are non-self. And so this beta helix structure, this blue structure, really screams non-self to the immune system. And so that's something that, again, we have some success already with exploiting that, and I think that's something we can do to exploit much more. Okay, I'm going to skip a little history of antibiotics because I think you probably know it. I'm going to skip this because I know I'm running low on time. And I'm going to finish with moving towards détente. So moving towards détente, how do we move towards détente? So what I've told you today is bacteria are not the enemy. Pathogens are, definitely, right? So we don't want to get rid of antibiotics. We need antibiotics to kill the bacteria who are trying to kill us. But we need to develop basically a more healthy relationship with bacteria in general. And because this is particularly important because while antibiotics can save lives, the current antibiotics that we use indiscriminately kill bacteria. So they'll kill any bacteria with, for example, a cell wall. And that's going to affect both the good bacteria in our gut, on our skin, in our belly buttons, as well as the pathogenic ones. Okay, so we've got to turn our attention to finding antibiotics that can selectively kill only the pathogens. So obviously I'm an autotransporter person, so I showed you a bunch of great examples from autotransporters. There's loads of other examples, right? That whole needle structure I showed you, that's something else that's very specific to pathogens, and you don't see it on good bacteria, and you certainly don't see it on human cells. Okay. So vaccines, I think, are a great way to avoid avoid the advantage that bacteria have to outstrip us in terms of cell replication. I think we should also think more about using protective barriers, right? So just washing your hands with soap doesn't have to be antimicrobial or antibacterial soap. Obviously, we can't all wrap ourselves in plastic wrap, but trying to separate us from the bacteria is good. And also, I think, trying to do more of this, exploiting this nutritional discouragement. Again, going back to that idea about our bodies already have evolved to use iron as a way to control the bacteria that can take up residence within us. Maybe there's other strategies along those lines that we can use. And along those lines, I think as we develop a better understanding of what the microbial flora is doing and how it works and how we can nurture it, then we can move towards some of these nutritional strategies. So I'll stop there, and thank you very much for your attention.